Hi, my name is Joanne Matsubara, and I'm a research scientist and faculty member here at University of British Columbia. My lab is in the Department of Ophthalmology at the Eye Care Center, and this is where my students and staff help me to run experiments in order to understand exactly why eye diseases occur in some people and novel ways to treat them. Our research focuses on retinal degeneration. One of the most common forms of retinal degeneration is age-related macular degeneration, or AMD. AMD affects about 12 million people in North America, and in its very late stages can cause vision loss and blindness. In the research labs, we study molecular pathways and mechanisms in order to understand how to prevent retinal cells dying in the diseased eye. We identify new genes and proteins that are highly overexpressed in the AMD eye, and once we identify these genes and proteins, we then find ways to suppress them in order to slow or even stop the development of the eye diseases.
Hi, I'm Wade. I'm a PhD student in the Cell and Developmental Biology program here at UBC. For my PhD research, I'm studying AMD and the interactions between two very important pathways, inflammation and cell death, using retinal pigment epithelial cells. We call those cells RPE cells, and they undergo atrophy or cell death in the dry form of AMD. One candidate gene is the X-linked inhibitor of apoptosis, or XIAP. This gene is very important as it protects cells from the apoptotic form of cell death. My work is focused on understanding how the XIAP gene interacts with the inflammatory pathways in the RPE cells. Inflammation is an important trigger that starts the disease process in AMD. The interactions between inflammasome activation and XIAP are not known, and we are among the first scientists to study this towards developing a new treatment strategy to prevent AMD.
All right, I'm Chris. I'm a PhD student here at the Eye Care Center, and I focus on stem cell and regenerative medicine therapies. Millions of people around the world suffer from retinal degeneration, and what we do here is that we try and take stem cells and we derive various retinal tissue from that. And then we transplant that tissue into the subretinal space, and this is in an effort to actually rebuild those lost layers and rebuild or recover visual function.